So now in our analysis workflow for clustering, we are almost ready to do the principal component analysis, but before that we need to scale the data and regress out unwanted variation. So in this video you will learn why we need to scale data prior to the PCA, how the scaling is done, and how we can remove unwanted sources of variation at this step. So ultimately we are going to do clustering and we will use principal components for that. So we'll need to do principal component analysis. But before that, it's necessary to standardize the expression values for each gene across all the cells, because this way, this will give equal weight then in downstream analysis so that the highly expressed genes do not dominate as they otherwise would. So in Chipster, this uses the scRNA tool, scRNA method scale data, which performs z-score normalization. So it shifts the expression values of each gene so that the mean expression across all the cells becomes 0, and it scales the expression values of each gene so that the variance across all the cells becomes 1. The scale data function has also an option to regress out unwanted sources of variation. So let's have a look at that in more detail. So there can be different kinds of sources of uninteresting variation. So some is technical noise, patch effects, and some is actually biological. So for example, cell cycle stage can affect expression, and unless we are particularly studying cell cycle, we might want to remove this effect so that the cells don't cluster based on cell cycle. So we want to remove this kind of variation, and the way how scRNA does it is that it constructs linear models which predict the gene expression based on a variable that you define. There can be different ones, for example, the number of detected transcripts per cell, mitochondrial transcript percentage, and so forth. So these variables are regressed individually against each gene, and then the resulting residuals are scaled and centered. And the scaled z-scored residuals of these models are then used for the PCA and clustering. So in Chipster, the number of detected transcripts per cell and the mitochondrial transcript percentage is regressed out automatically. And then you can choose if you also want to regress out the cell cycle effects. There is a separate video on that. And again, this happens in the same tool that does a lot of other things. The parameter is at the bottom here. So here it's whether you want to regress out the cell cycle differences or not.